The first antibacterial-resistant superbugs were the MRSA, which were found in 1960. The amount of antibacterial-resistant superbugs after that were increasing with the amount of antibacterial produced. Antibacterial-resistant bacteria can cause death and destroy the economy of infected patients and the hospital that are housing those patients.
Just like any living organisms, bacteria wants to survive and reproduce as best as it can, mostly through a process of genetic mutation, which happens through a short period of time or a long period of time, depending on the variation of bacteria. Usually, these genes are antibiotic resistant because antibiotics pose one of the biggest threats to bacteria, since it kills almost all of the bacteria itself. There are several ways a bacteria can remove antibiotics. One of those ways is through an efflux pump. An efflux pump actively pumps out antibiotics from inside the cell to outside the cell. And another way is through molecular bypass. Usually through a synthesis of many mechanisms together, it provides the best chance of survival and the best adaptability and the best survival rate for these bacteria. One main method of how these genes spread is through bacterial conjugation or horizontal gene transfer. This process is when two cytoplasms connect to each other, and then the plasmid DNA from the original one gets transferred to the copy one, the clone one. After that, it gets replicated, and then both bacteria will have that AMR gene.
The CDC, Center for Disease Control and Prevention, has classified superbugs into 4 categories: urgent threat, serious threat, concerning threat, and watch list. In order to prevent a potential disaster from happening in the future, CDC is putting special effort to monitor superbugs that belongs to the category of urgent threat and serious threat. 2 bacteria-resistant pathogens that are in urgent threat are CRE and Neisseria gonorrhea. CRE is a highly antibiotic-resistant bacteria that cause severe infections and has a mortality rate of up to 78%. Neisseria gonorrhea is a bacteria that has afflicted humans for centuries, causing infection to reproductive organs. Antibiotics that are used to treat these pathogens are no longer effective. They have gained resistance to it, and new antibiotics are still in the process of testing.
A few solutions for antibacterial resistant bugs are phage therapy and reducing the usage of antibacterial. Phage therapy is using phage to destroy the superbugs, and by limiting the amount of usage of antibacterial, we can lessen the chance that the bacteria will adapt and mutate to counter the effects of antibacterial. Other organizations such as WHO and CDC are finding ways to help gather more information about antibacterial resistance. For example, WHO is developing a program called GLASS, which can help them gather more information about bacterial and how to counter them.
The purpose of this report is to increase awareness and knowledge of AMR. We want to increase public awareness of AMR since it is a massive problem worldwide, and the effects are catastrophic if left at the same rate that is going right now. Most people aren't aware of AMR, and people are usually prescribing themselves way too much antibiotics, or using antibiotics as a miracle to solve any drugs, which is not the case. It only makes them worse and create more superbugs and more antibiotic resistant bugs, which will harm them in any way and harm society in any way. We also want to encourage scientists and funders to put more money and put more scientists on this case, since it's an important topic worldwide with very serious effects.